I don't wanna grow up, I'm a curious kid. The world has 100 questions I can play with. So I'll open my arms and eyes and wonder every day till the day I die. No one really knows why gravity exists.
All right, well, thank you, Tucker. So although you heard about my background, it's neuroscience, human evolution, and so on. Today I'm going to wander away from each of those. You're welcome to ask me questions about the nervous system, how brains develop, how they've evolved. My background includes studies of things like fetal neural transplantation and things like that. So I'm that's my background, but I've become very much interested in a problem in evolutionary theory. And it's a problem that's been around really since Darwin's time that's never been really dealt with. And so I call it inverse Darwinism, and I'll try to explain what that means. It's not anti-Darwinism. Clearly what I'm going to be talking about is right in Darwin's kin, but it's in fact something that he didn't think of. And I think he didn't think of it in part because there are so many things he didn't know. Didn't know how reproduction worked, didn't know how physiology worked, and didn't even understand cell theory at the time. Obviously, all of this stuff has happened in the last many, many decades since then. And in fact, I'm going to suggest that the part I'm talking about really doesn't start to gel until the late 1990s. Although, as I'll mention in a moment, it begins really in the 1970s, this view. Inverse Darwinism does not invert most of Darwinism. It inverts one thing, and that is the concept of selection, which in many respects people think is all that Darwin is talking about. I'm going to argue that in fact it's not, and that in fact it fits well within Darwin's story. So let me begin at the end. This is the last line of Darwin's Origin of Species. There's grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one. In some various versions of this, he throws the creator in here, but in this version, which is actually the first version, he does not. And that, whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Many people quote this last part about how beautiful and wonderful and exciting natural selection is. I bring this out for one very simple reason. Darwin did not like to talk about the origins of life. In fact, when people even asked him to, he avoided it. Once in a while, in a letter to a friend, he might say something about it. But I want you to look at the part that I've highlighted in red here. He says that with its several powers, he's talking about life, has several powers. And what he's saying here, in effect, is why he doesn't want to talk about the origins of life. The origins of life is not a problem of natural selection. That is, natural selection, all of Darwin's mechanisms, assumed several powers. What life does. It doesn't talk about how life came about, although we're struggling to do that in my own work. I'm interested in it as well. I won't talk about that today. But there's something more. And so what I want to talk about is one of those several powers, and why we've ignored it, and why we've been, to some extent, captured by Darwin's original insight without recognizing its broader implications. So I'm going to go back to 1838. Darwin is writing in his notebook after chatting with a few people that were talking about raising beef cattle for marbled meat, and Darwin was a little bit confused by this. He goes back and he's reading this book by Thomas Malthus on population. Malthus is often called the dismal philosopher because what he basically said is that, look, I'm looking at the way that the population of England has expanded so rapidly and noticing that agriculture is not keeping up. That is, the support for people is not keeping up with the reproduction rate of it. A very good analysis he was doing of what was going on at the time. And he said, as a result, we're in trouble. By the end of the century, things are going to be horrible. And so a lot of people sort of looked at this and worried about it and so on. It turns out that Malthus was of course wrong because of course industry comes in, coal comes in, a whole bunch of things change this. But we're still struggling with this question of Malthus. But it's really, Darwin himself says this after reading Malthus. He sits down and reads Malthus and then he has this epiphany. And he says, aha, 3 ideas will explain it all. He's been struggling with this idea of evolution for a couple of decades now. And these are these 3 ideas. This is directly from his 1838 notebook. He says these 3 ideas are going to explain it all. Grandchildren like grandfathers. Inheritance, right? Inherit traits. But tendency to small change, especially with physical change. So he's talking about the variation, that your kids are varied. They don't have exactly the same traits. And then he says, and great fertility in proportion to support of parents. That's the Malthusian piece. That was what Darwin felt. And he writes this in the preface of The Origin of Species. That is, this reading of Malthus had him realize that if this is the case, then if there's going to be variation, and there's going to be limitation, there's going to be battling over this, and those that succeed, that fit best with their environment, that make best use of the features that are there, they'll reproduce, the others won't. They'll be selected out, natural selection, sort of a negative view of selective breeding from Darwin's point of view. That what you get left is those that fit well, and the rest don't make it. What I want to do is to say, all of this is right, but Darwin didn't realize that the Malthusian story is not the whole story. And it's that little bit that I'm going to invert, in which it's not that there's great fertility in proportion to support, but in fact, sometimes there's more support. And if there's more support, that's maybe a good thing. It will change the whole dynamic. To draw this forward, let me just sort of give you a kind of a diagram with Darwinism on one side and inverse Darwinism on the other side. So Darwin, the first 3 principles: inheritance, inheriting traits, variation of that inheritance, and then overproduction. His argument is that if there is, shown in red here, a basically limited resource, some are going to make it, some aren't going to make it. And as a result, you're going to get adaptation. By that, what he's saying is that by simply removing those that don't fit so well, if there's not enough to support everybody, to support everybody's reproduction, those that do succeed oftentimes are well fitted to their environment in various ways. That's what he's trying to explain. Adaptation. How are things so well fitted to their environment? This is a process that immediately people grasped. And as a result, realized that this would help explain what looked like this difficult teleological question about evolution. Why do things work so well? Why does everything fit together so well? However, what might happen? How might we think about a situation where there's not a resource constraint? What examples might we take from this? Think about your own body and its development. Early in development, you start out as a single cell. And that single cell duplicated inside of a very supportive environment. There was no competition between these cells. They're growing and growing and growing. Everything is great. It's not Darwin's situation. However, each cell does inherit genes from the previous cells. They will vary over time by where they're located, by what signals they get, and so on. So inheritance and variation are there, but in the development of a body, you don't have this one problem. So I want to broaden these concepts out a little bit because it's a thermodynamics story he's talking about. So the first part of it, of course, is inheritance, duplication, producing more and more cells. There's a kind of redundancy there. In fact, we now know that if you pluck a cell out of the developing embryo, oftentimes it'll be replaced by another one. Or if a whole group of cells are mixed up, for example, in twins that get mixed up in early embryology, the cells just sort of work on together. They don't show up. So if you have a twin, it's possible that you and your twin might share some cells in common. That one person might also be the result of two twins that have been consolidated. What's going on here is there's not selection, there's not competition. It does turn out that during development, as organisms get more complex, competition does play a significant role. So in the development of the brain, an area that I'm very much interested in, the fine-tuning of things is the result of competition between cells and connections. And as a result, brains are adapting. They're adapting to the body they find themselves in. They're just a bunch of cells initially, but they have to adapt to the body they find themselves in. And they actually use a kind of a Darwinian process. In fact, someone actually called this neural Darwinism years ago. So the Darwinian model about adapting and fitting works even at the level of development. And we'll find out that it works, in fact, inside of cells as well. I won't go into all of those details. But the point I want to make here is that the Darwinian story, if we just sort of invert that last piece, has other implications. That maybe the idea of evolution, which now can be linked to development to show that they actually share something in common, now you can actually say something else. So what about variation? What's variation? Variation ultimately is just the second law of thermodynamics. Things get messed up, errors get made, and they accumulate over time. You just can't resist it. It just will keep going, keep going. And so what happens, and we'll see this at the genetic level, but it happens at all levels, that the increase of entropy is always there. The increase of entropy will turn out in the story I'm about to tell you to be a very important and useful feature. And then third, what about overproduction? Well, that's what's going on when cells multiply as your body develops. But it happens in all kinds of situations. And I'll show this happens at the level of the genome as well, at lots of levels. And to understand this is to understand both evolution and development, but also how evolution is more complicated than we thought it was. The key to this is that I'm going to show that by thinking about it in inverse terms, I'm going to have to sort of move this up a little bit. There we go. Keeps falling down. I want to show that, in fact, in this reverse approach, one of the things that we see is that things get more complex. In fact, the explanation for complexity in evolution turns out not to be the result of natural selection, but actually of its inverse. And that's what I'm going to try to suggest to you today. So here's a little sort of common sense way to think about it. This is a quote from Plato's Phaedrus. This is Socrates talking to somebody else. And Socrates is talking about the problem of the development of writing. Writing is going to be a problem, he thinks. And here's what he says. He says, if men learn this, reading and writing, it will implant forgetfulness in their souls. They will cease to exercise memory because they rely on that which is written, calling things to remembrance no longer from within themselves, but by means of external marks. Notice what I have on the right side of this. How many of you remember all the phone numbers that you used to know? Right. This is something that naturally happens. If you've got support, if somebody's giving you a free ride, and they do it again and again, or the device makes this possible again and again and again, things degrade. In fact, the degradation is just as a variant of the second law of thermodynamics, of entropy increasing. You know, we don't have to do much, we do less work and less work and less work, and we can't do it after a while. I'm actually going to argue that this process is a critical process behind the complexification of evolution, how things get more complicated. Notice that they get more complicated, as I'm claiming here, precisely because they lose details. You've got redundancy. You know, this is a backup. I don't need to remember all this stuff. The problem, of course, is that you gotta remember your phone. So I'm going to start by giving you some examples of this kind of process in evolution. First of all, at a very basic level, and then try to work my way up. So the question that probably began this search, it was a question that begins in the early 1900s, is once we figured out that there was genetics that was responsible for organisms and passing on information, a question arose. Well, where do new genes come from? How do they show up? The neo-Darwinian theory says, well, old genes just get damaged, and maybe you get a better damage, and you can use it. The problem is, that's not a new gene, that's a modified gene. Where does a new gene come from? This was a problem. There are various hints at it, but of course we didn't know quite what genes were until the 1950s. What happens is that, in fact, we began to discover that there were some issues about this, because we had at least one clear example that people knew about quite early on in the 1900s, and that's trisomy 21. That is Down syndrome. We know that if you duplicate a whole chromosome, you've got 3 copies of them. Trisomy means 3 copies of chromosome 21. You've duplicated all of the genes on that. You've got too many of them. It turns out that having too many genes, having redundant genes, can be bad news. It can cause difficulties. And we know that we can't have too many other trisomies. Trisomy 8 is one of the other ones that can be survivable. Most trisomies are deadly. Most times you duplicate genes of a whole chromosome, it's really a problem. There is one special case that half of you in the audience, roughly, have to deal with, and that's the X chromosome. I have only one of them, but many of you women have 2 of them. In fact, I suspect most of you do. Interestingly enough, you only have one expressing itself in any particular cell. The others, because it's so dangerous to have so many genes duplicated, it becomes what we call a bar body. A bar body basically is an inactivated chromosome. The inactivation is very interesting, and the man whose picture I show here is one of the people who originally demonstrated this. But he did something else, and that's what I want to talk about. Although people have recognized that there are some cases of gene duplication, where we saw genes being just copied next to each other, like the duplicated chromosome has genes copied on it. He recognized that, in fact, we're beginning to see gene duplication of a very massive sort. This guy's name is Susumu Ono, and he wrote a book in 1970 called Evolution by Gene Duplication. He's trying to answer the question, where do new genes come from? Well, he was now, again, we're looking at some 20 years after the discovery of DNA, people were beginning to measure amounts of DNA. And one of the things that they had found is that there are lots of plants that look like they have double the amount of DNA that they should. In fact, if you like strawberries, they have typically 8 duplications that have taken place. Their entire genome has been duplicated a bunch of times. Wheat, a couple of times. So he felt that maybe this was the answer to how new genes show up in the world. So here's a couple of diagrams. Let's see if you can. I gotta use this one. Okay. Let's see. This is the. Here we go. So on this side, this is a phylogenetic tree, evolutionary tree. So this is an ancestor of chordates. Chordates are all these creatures that have this sort of backbone-like thing, even though it's not always a backbone. And we see in the inheritance, as we go down the line, here's birds and mammals. Here's basically land vertebrates, all land vertebrates, amniotes they're called. Some fish here, euteleos, and then the teleos down here. These are like salmons and trout and so on. It turns out that of each of these X's, 3 X's, is a place where, in the course of evolution, the whole genome, every single gene, got duplicated. And it happened again. So in the lineage that leads to us, there are 2 whole places where the whole genome gets duplicated. So Susumu Ono was realizing that this may be the way to deal with this problem of where new genetic information comes from. It turns out it's not that simple, and I'll try to get to that in a minute. Interestingly enough, so that we've had 2 duplications that led to us. This group of fish, the teleos, down here, these ray fin fish, had yet another one. So they have basically 6 copies of each chromosome. An amazing feature. It turns out that this is not necessarily a good thing. And you notice it hasn't had a lot. It happens a few times over hundreds of millions of years. So it's a rare event. And the reason it's rare is just like the trisomy 21 story, there's lots of bad news when this happens. Interestingly enough, in plants, shown here on the right, it's happened multiple times. Also, in these early phases, you get two major duplication events leading to all of these plants. Gymnosperms here, only one of them, so for example, pine trees and things like that have half as many as other flowering plants do. They've gone through another genome duplication step, but then you see a lot of small ones I showed here. Get this so you can see them here. These are duplication steps. So there's like 4 duplication steps leading to sorghum and so on. There's a couple more. There's 5 leading up here to the top, the papyrus, to papaya. It turns out that gene duplication is more of a regular thing in plants, and as I mentioned before, a number of plants like strawberries have gone through a number of other genome duplication events. So, first of all, the answer to this starts to look like, oh, you just duplicate the whole thing, you got a lot of new stuff to play with. The problem is, you're overproducing a whole lot of things, it's no longer coordinated, and if you think about trisomy 1 being a problem, whole genome being duplicated, this is a serious problem. Now, there's another way that genes get copied. And this is sometimes called transposons. I won't go into the details of this. But what it amounts to is that there's a part of the genome in which it literally cuts a gene out and splices it somewhere else. It's like cut and paste in a computer. This happens a lot. And it turns out that it can happen in a very helpful way. In the case of this one, amylase. Amylase is basically a sugar, a starch sugar that you find in grains. And it turns out that animals that eat grains have actually duplicated this gene multiple times. And what that does is it's a dosage effect. It just makes it so that we produce a lot more. In fact, the big duplication events that have taken place like this have to do with the basic machinery of building cells. So that the genes for what we call transfer RNA, for RNA molecules that are involved in a structure called the ribosome. These are things that every cell has hundreds of these, and then generally turns out to be maybe hundreds of those genes just to produce enough of it to keep up. So one thing you want to do, if you can increase dosage to your advantage as opposed to your disadvantage, then just duplicate it. So what we see in humans, show here at the top, particularly populations, it turns out there's different, even in different populations. Populations that are eating more grain have this gene duplicated many times. It's actually an enzyme that's produced in saliva. But you can see other animals, and typically animals, like mice and so on that hang out with us, also turn out to have a lot of this stuff. Granivores do a lot of this. Now, gene duplication is something else that he was focusing on, and realized that whole genome duplication may be a problem, but individual gene duplication might be more helpful. And I'll look at a bunch of examples of this. Notice that when we talk about gene duplication, what we're talking about is that a particular gene gets copied, cut, pasted next to itself, or pasted somewhere else in the genome. This is a process that happens all the time. It turns out that a vast amount of our genome, more than many other species, is of this duplicated genome stuff. We've got lots of transposons, these particular gene segments that copy and paste things around. Now here's what happens. Sometimes they get pasted right next to each other, so you got a whole line of them right next to each other. Sometimes they get tossed off on other chromosomes. It turns out that this is a pretty common feature. But notice, now go back to what I started with. Here we've got, in a sense, something like replication, duplication. And now what happens when you've got multiples? Well, now you've got redundancy. One of the reasons we oftentimes, put a key out under the rock outside of our house is we need a redundancy, because sometimes things go wrong. I might have lost my key, but I can still get in. The good news about having redundant copies of things is that, in fact, it's a backup. You know, those of you that are using computers know that you gotta have backups. Periodically things can go wrong. Best to have redundancy. Well, life produces redundancy. It's just what life does as one of the several powers. One of the several powers precisely because life has to stay ahead of the second law of thermodynamics. The best way to do it is to overproduce just a little bit, to have a little extra if things go wrong. Well, now let me talk about another variant of this story. This has to do with smell. To smell is the most genome-demanding sense. Why? Because we've got to have molecules that match other molecules that are floating around in the air. But to really sense the complexity of the molecules in the air, we've got to have a lot of them. So it turns out that there's a huge amount of gene duplication at the level of what we call olfactory receptor genes. These are genes that produce a protein that actually binds to a particular odorant. But you've got to have a lot of them. So it turns out that the duplication of these genes is quite extensive. But the second thing that's extensive, and this is what's going to be important for the rest of the story, is that the genes now have undergone a little bit of degradation. We might call it variation, but each of these copies can now be just a little bit degraded from each other. None of them are exactly alike. And some of them are what we call pseudogenes. Pseudogenes are that variant that, in fact, shown here in blue, that no longer do anything. They were just duplicated, but they became noisy so fast that they stopped working. If you're duplicating things a lot, and you're allowing the duplicates to sort of wander off and do whatever they want because they're redundant, there's going to be a good chance that they're going to just sort of wander off to uselessness. It turns out that that's what's happened. But life puts up with this. Life is something that is very productive, proliferative. It's exactly what Darwin was worried about. It's that proliferation, that overproliferation. But if there's no real cost to it, there's no competition, then so what? So what we see here is that elephants, not a surprise, have the most. Notice that there's close to 2,000 variants of olfactory receptor genes. This is a gene that got duplicated, got duplicated. It's now all over, and all of the chromosomes actually have some of these. And they're all variants of each other. They've all randomly varied. But having a huge random variation is one way to capture the range of possible odorants that are out there. This is also the logic of our immune system as well. Our immune system also generates multiple copies of these immunoglobulins, these things that are going to bind to other cells. But what it does is it does what we call hypermutation. It generates it and then modifies it, generates it and modifies it. So those genes are also modified in this respect. That's because the system has to have enough variation to capture all the possible variations that might come in with viruses and bacteria and so on. So pseudogenesis, we sometimes call this, that is producing pseudogenes, is actually what happens most of the time. Most of the time when these duplications take place, they just wander off to uselessness. And we know what they are. We recognize pseudogenes because we can look in the genome now and see that it has the structure of a gene that we know does work or worked in some other species, but somehow it's not being expressed or it's full of noise in some respects. It's got all the right features. It's like, the right story is told there, but then there's a bunch of randomness in it. So we know it's a pseudogene. It's a false gene. But that means it's kind of you might say, it's a fossil of a once useful gene. But what happens because of this, and this is a variant of what I was saying about the olfactory system, is that by virtue of this duplication and variation, new combinations are possible, in which one function now becomes subdivided into partial functions. There's a simple sense of this in which one gene duplicated itself and one of them begins to lose its sensitivity. But if having too much of that protein produced from that gene is a problem, what can sometimes happen is the second gene loses its sensitivity. And now that the right amount of protein is produced by 2 genes instead of 1. But now you can't lose one of those because now you won't have anything produced. They've become effectively addicted to each other. You've got to have both of them now. So one of the things that will happen is that whenever there's not a function that has to be maintained, it'll spontaneously degrade. Degradation is the rule. But what happens because of this is that now they can become related to each other, linked to each other. My favorite example of this is a very common molecule that probably all of you know about, hemoglobin. It's that protein in your blood that captures oxygen and moves it to the rest of your body. Hemoglobin, actually this is the evolutionary tree of hemoglobin. And what we see here is that the ancestor, way back here, was the ancestor to hemoglobin, but also to myoglobin. What's myoglobin? It's the similar protein in your muscle that makes your muscle look red. Why does hemoglobin look red? It's because it's rusted. It's captured oxygen. It's iron that's captured oxygen. Makes it red. Well, it turns out that early on in the course of chordate evolution, one of the things that happened is that gene duplicated. Now, the duplication event produced one that's inside of cells, myoglobin, but now one that floats around in blood. One of the things that happens oftentimes with a duplication, and this turns out to be a very important feature, is that the duplication process will produce a problem of dosage unless the 2 genes are turned on or off in different places. In this case, there's no dosage effect because one of them is floating around in blood and one of them is in your cells, your muscle cells. But that means that your blood can pick up oxygen from your lungs and then give it up to the myoglobin in your cells. So it's a wonderful 2-step process. Now they have become, in a sense, dependent on each other. By simply varying how they're expressed and how they grab oxygen or give up oxygen, you now can have a relationship in which there's a new synergy that's been created. But it turns out that it doesn't stop there. Because one of the things that happened is the hemoglobin in your blood is not just one protein. It's in fact 4 proteins all stuck together. What we call a tetramer, 4-part molecule. They're not fused to each other tightly, loosely, and so this picture I showed out here is the one we typically find in blood. It's a 4-part molecule. 2 green ones, 2 blue ones, and they each hold an iron atom, and the iron atom is what captures the oxygen and can give it up to the iron atom in myoglobin. But it turns out, interestingly enough, that it didn't stop at that. So one of the things that clearly happened is it got duplicated initially, and we get the blue group up here, the alpha and the zeta group, and down here in the green, we get the beta group, which turns out to be more than just beta, as I'll show you in a minute. And it turns out that as each of these genes modified, it was the part of the protein that's on the back side, away from the iron, that modified in such a way that they just tend to stick together like hand in glove. And so they just tend to hold together into a 4-part. But that means it can now hold much more oxygen. Now this little molecule floating around holds much more, but also because the iron atoms are somewhat closer to each other, it's a little weaker and it gives it off more effectively to the myoglobin. So rather than having just one hemoglobin here now, 4 of them come together and they begin to cooperate. They don't want to cooperate, it's just by chance they got stuck together. But if they get stuck together, now natural selection comes back to work and says, well, this is a good deal. We're going to keep it, and those that don't get to stick together, we're not going to keep. Well, it turns out that it even gets more complicated. As I show down here in the green, the beta chain also duplicated multiple times. So what are all they doing? It turns out that they only do something useful in placental mammals like you and I. So here I show these up here, the beta and the alpha, but the beta is changed here with gamma. So get this doesn't fall down into my chin. What we see here is that by duplicating the beta, here it is out here, multiple times. This is a chunk of DNA stringing along at the DNA. There's a region. This is a region that's one of the gammas, the G gamma and the A gamma, and then there's the delta and the beta. Beta is the one that's in you and I. But during gestation, beta is produced. Epsilon is produced initially. Here we see its value at the very earliest stage of the yolk sac, dropping down in quantity. Then the gammas begin to show up. So during gestation, there's a different hemoglobin produced. Then as you're about to give birth, that one is shut down and beta comes up again. What's going on here? The problem, of course, is the fetus needs to get its oxygen from mother's hemoglobin. How do you get oxygen from mother's hemoglobin? You've got to have a stronger grasp of that molecule to be able to take it from mother's hemoglobin into your own hemoglobin. But now you've got the problem. You've got to give it off to your own myoglobin. So you've got to have two different strengths here. So it's an intermediate value. By simply duplicating the hemoglobin beta gene, it made internal gestation possible. It's because now the fetus can go through phases in which, as it grows and gets larger with different oxygen demands, it's able to get it from mom. But as soon as you get out, it's not an advantage. And so it turns out it's shut down and the beta shows up. So this is an example of a kind of synergy that's developed just by this, you might say, odd chance issue of overproducing something, duplicating it, by redundancy, allowing it to degrade a little bit. And then, by virtue of that, combinations are getting tested. One of my other favorite examples has to do with the fact that you and I, or most of us, see in three colors. Most mammals don't. Monkeys and apes do. And in fact, some New World monkeys do, but many New World monkeys don't either. And here's a sort of evolutionary tree that shows the development of that process. The guys over here in red are the ones that see in three color, like you and I. We call trichromats. And the others, some of these, dichromats, mice on the far end, and some New World monkeys also are dichromats. That's most mammals. That means there's two genes for color receptors. And these genes are called opsins. Opsins produce this pigment-like protein inside of the cones of your eye that allow that cell to pick up a certain frequency of light. What's happened is that there was a duplication event. Only in monkeys and apes, and particularly in the New World, that produced this trichromatic vision, which you can see in all the colors we see. How did this come about? Well, it's the same story. It's a gene duplication story. As I'll tell you in a few minutes, it's a somewhat more complicated gene duplication story, but an interesting one at that. So what's happened here is that actually there were two color receptors you see at the top, dichromat. The sensitivity for color is represented by this little bell curve here of the two opsins. They produce a protein that picks up different colors. Well, it turns out that the green one, what we call the low frequency one, the L here, long wave, low frequency, was duplicated. And as it duplicated, the duplicate slightly changed its function. It lost a little bit of sensitivity and wandered towards the red. And as a result, now we've got, and you can see this is a picture of the gene duplication process down here at the bottom. We're now capable of seeing in three colors because of this. Yeah?
First, do you mind questions?
No, please, if I'm going too fast or skipping over too much, stop me.
I have two questions. First, the evidence of tricolor vision in animals with whom we can't communicate very well is the number or the variety of cones, whether they have the three kinds of
three
cones?
kinds of cones versus two kinds.
Okay. And then you mentioned something about the evidence of the three-color evolution, three-cone evolution, happening most evidently in the New World. But I thought maybe
the Old World.
Old
World. There are three copies of that gene in the New World, but only some New World monkeys actually exhibited. So let me just show you this again. So here, New World monkeys are on the left, primates from the Americas. And you can see there's just one place right here where we get a gene duplication event. And so there's some of these monkeys that actually can see in 3 colors, but most of them can't. Mostly it's just in the old world. But it turns out that it happened independently in both these 2 realms. So one of the questions is, why not other mammals? And it turns out that I'll try to come back to that with another gene duplication story. But this is actually not a gene duplication story, but actually a story about the loss of gene function. Primates, okay? One of the things about you and I is that we need vitamin C. We need to eat food with vitamin C. Magellan sails around the world and loses half of his crew to scurvy because we need vitamin C. All old and New World monkeys need vitamin C. But New World monkeys have some adaptations that we don't have. But all of us Old World monkeys and apes, like us, need to eat vitamin C on a regular basis. And so I show this diagram here, which is, that shows that, there we go. These red areas, these are the groups, the chimpanzee and other monkeys, Old World monkeys here, old OWN. New World monkeys, a few of them, have picked up 3-color vision. There's a few cases where we see this. Many birds see in 3 colors. In fact, the dinosaurs, as we figured, probably all were 3-color vision animals. The ancestors of mammals, however, almost certainly were nocturnal during the age of the dinosaurs. Nocturnal vision, you want to have broadest sensitivity to light, to take in as much light as possible. To see in a narrow color range, you have to give up some light sensitivity. That's why when it's dark, we don't see well in color. Well, it turns out that in most mammals, it stayed that way. Most mammals have never needed to go in this direction. For some reason, primates did. In fact, mostly just a small group. Now, there are a few other animals, guinea pigs, curiously, like that. There's a few other animals as well. Fruit bats, and as I said, many birds. Fish sometimes have 4-color vision. So it's not something special. It's actually, we came from a sort of impoverished group, this nocturnal adaptation that we had. So what happened actually here? Well, what happened is it had to do with fruit. Now, here's the problem. If it had to do with fruit, we didn't have to eat fruit, did we? Couldn't we eat something else? Well, it turns out that back in 1994, a group of Japanese researchers who were looking at genes of various kinds, asking this question, is there some genetic story to be told here? They found in a rat that rats produce a protein produced from a gene called GULO, or LGO. You can see the complicated name here, L-guanyl gamma lactone oxidase. Most people call it GULO these days. They found a gene in the rat that makes ascorbic acid. Rats make their own ascorbic acid. Dogs make their own ascorbic acid. Cats make their own ascorbic acid. But you and I don't. One of the first things they did, because of the technology that they had, is they marked with a radioactive tracer the rat gene. And they used that as a probe, first of all, in us. And it turns out that we have a copy, but it's full of noise. We have a pseudogene for making vitamin C. A gene that has many of the features of it. And here I show a little short chunk of it. These little parts are what we call exons, little parts of that gene from the rat. And here's the human. And each of these lines is a mutation, is a damage. In fact, the damage has a little stop sign down here. That if that gene is being read, that says stop reading at this point. So it's useless, doesn't do anything. In fact, one of the things that happened is that it occurred now off to the left of this feature. There's another major mutation that happened. It's called a frame shift. You remember the old film strips that had individual pictures, and if it got slipped out of position, you saw two halves of a picture, that's a frame shift. Well, it turns out that genes are read by 3 steps each, what we call 3 nucleotides, or 3 base pairs. And every part of a protein, an amino acid, is coded by one of these 3. But if one is knocked out, it shows everything off a little bit. So the whole gene becomes nonsense. That's what happened to the gene that we have. And it turns out by looking at the noise that's accumulated in all the other monkeys and apes that have this problem, we can actually get a sense of when it happened. What happened is that somewhere around 60 million years ago, way back, primates had a normal functioning ascorbic acid producing gene. But something else happened at this time, and this is known now from the fossil record as well. And that is, they were hunting at night. They were mostly eating insects and small vertebrates like frogs and so on in the trees. But what we see at about the same time is that nocturnal animals, which have really big eyes and take in all the light they can, their eyes tend to get smaller. And their teeth, which were really good at eating insects, which means they're sharp and they chopped up the exoskeleton of the insect to eat it, now became more flattened. So we know that what was happening is that they were beginning to now forage during the daylight. There's more light available all of a sudden. And they're having a different kind of food. What were they doing before this? They were hunting small insects and vertebrates in the trees. But there was another food source in the trees available to them. Fruit. Fruit was originally, we think, evolved for birds to eat the fruit and to poop out the seed somewhere else. Do good seed dispersal. But primates living in the trees found this food source, full of sugar and stuff. Well, that's great. We're going to eat this. And in the process, they were getting lots of vitamin C. So the story is, over time, what happened is that as vitamin C was being produced, it was like having a gene that was already producing it. If you're getting vitamin C every day, it doesn't matter whether your gene is working or not. And so, the second law of thermodynamics, things just begin to break down, get full of noise. And so, in the course of the tens of millions of years since this started, the genes in each of these lineages degraded in different ways. But now, we're addicted to vitamin C. If we don't get it, we get sick. So, this is another variant. It's not like in the gene duplication where it's inside the body and you got redundancy, but now it's redundancy outside the body. But that means you've got to keep it. You can't let it go. That links us back to the last question I was talking about, 3-color vision. Birds have always, as far as we know, had 3-color vision. Fruit needed to get birds to disperse its seeds. How did it do that? Well, it got to do a couple of things. First of all, you've got to wait till the seeds are right. If they disperse them too early, they'll die. They won't germinate. And if you don't have something for the birds to eat, sugar, they're not going to go for it anyway. So now the plant has a problem. The plant's problem is this. I've got to keep birds from eating the fruit and dispersing my seeds until it's just right. So how do I advertise for the birds that it's right? I want to hide it from the birds in the leaves. Keep it green, keep it green, keep it green. Now it's okay. Make a color change. Okay, but if you don't have 3-color vision, you can't see it. The color change is typically from green to something of a longer wavelength, like red or orange or yellow. So in effect, now we have a story that allowed us to link back evolutionarily and to see that this change in the structure of the eyes and the teeth of the skulls of these ancient primates, and the change in the gene that causes three-color vision, and the loss of the function of the gene that produces ascorbic acid, all seem to go back to about the same time. They're all linked together. Remarkable find, but interestingly enough, it's even more interesting than that, because the fruit had to keep its sugar good while it's maturing long enough for the seeds to get ready to be germinated so that birds would like it and eventually primates would like it. How do you do that? You've got to protect it from oxygen. How do you protect fruit from oxygen? With an antioxidant. What better antioxidant is there but vitamin C, it turns out. Slice your apple open, leave it out on the table, it'll turn brown. It's got oxidized. Fruit figured out a way to protect the sugar inside of it for a long period of time so that birds could get the sugar and therefore disperse the seeds appropriately. A complicated evolutionary synergy was developed here, including a kind of addiction that we have to this stuff, all because of this collective need. It turns out, as a result, not only do primates have this extra vision, but hey, we can also find sweet things and sour things interesting. Your dog doesn't. Our taste receptors have changed also to make it so we get this stuff. Because if we're addicted, we've got to get it. So I want to close up. I've got just a few more minutes here. I want to close up by talking about this process and how it's relevant. And ultimately, hopefully, say a few things about us and why it actually is relevant to who we are, not just that we need vitamin C. This is a case of a similar sort. This is a case of aphids. Many of you who grow plants find aphids to be a problem. Aphids are interesting because they have this ability to penetrate deep into the stems of plants and into leaves, into the phloem and xylem, the sugars that are being passed up and down the stems and so on to keep the leaves alive. They can do so because they have a little bacterium in their stomach, shown here in this little blue stuff. And what it does is the bacteria produce an enzyme that allows them to sort of burrow into the plant to in a sense, acid their way in, and then suck out this. So if the aphid didn't have this, it couldn't actually survive. So the aphid has in its gut bacteria that are necessary for it to feed. Just like we have bacteria in our guts, maybe for different reasons, of course. What I show you on the right is something interesting. We see that this bacteria has cousins that are free living. The cousin's genome is sort of indicated here by all of these little color spots. These are all the coding parts of that genome, the parts that produce proteins. This inner one is the genome of this little bacteria. By being inside of the aphid, and the aphid actually creates a nice little place for it to live and feeds it and so on, because the aphid is taking care of it, its genome is degraded. It's lost roughly half of its genome. This is the case of most parasites. They're simplified. They're simplified because they can take advantage of somebody else. Well, it turns out that this is a really general principle as well. One of the things we know about the cells in your body and the bodies of plants is that they have little organelles. So in all multicelled animals like you and I and plants and so on, we have mitochondria, these little tiny structures that turn out to have evolved from bacteria. I won't go into their independent background, but they became, we think, either parasites or maybe even like food or living inside of cells. So all of your cells have a mitochondria. The mitochondria does something really useful. It turns oxygen into energy. It turns out that in early life, oxygen was bad news. In fact, when photosynthesis developed, it poisoned the planet with oxygen. And a whole lot of animals, there's a huge extinction event. But there are a number of bacteria that learn to use oxygen. And it turns out that complicated cells, eukaryotic cells, all complicated cells, have incorporated these bacteria in them that utilize oxygen. So it's no longer poison, it's actually useful to provide energy. So this is a nice cooperative relationship that developed in plants. Yet have another one, chloroplasts, which also developed from a different kind of bacteria. These are bacteria that were doing photosynthesis, that are also inside of plants. So the plants are sort of this collection of two kinds of bacteria inside of a cell. Well, it turns out that the same problem develops here. And that is, if you become inside of something, what we call an endosymbiont, you can take advantage of this bigger cell. And as a result, what we find is that mitochondria on the left, now incorporated into a cell, and chloroplasts on the right, incorporated into a cell, have lost most of their genome. By being inside of these cells, the cells are providing so much that there's no selection for losing genes. So all but the most essential genes are lost, and so what you can see is there's only a 1% of the genome is lost in mitochondria from the original bacteria. Only 2% are retained in chloroplasts. Most of the other genes are gone. And not only that, some of these little arrows show that some of the genes have actually migrated into the nucleus of the cell, so that the cell actually produces something that the mitochondria needs. The mitochondria produces something the cell needs. They are now addicted to each other. This is a sort of variant on this last story. But now, each of these stories I'm telling you is about how cooperation develops. How, not by forcing things together, but by the accident of things coming together in an environment where they can provide excess of something. They can now give up a little of their function. They can become a little less autonomous and eventually, like in this case, totally addicted, totally parasitic, but they're no longer parasitic because they provide something of use. That's a new synergy is developed. This is about complexification, how things have become more and more complex, not by simply Darwinian mechanisms in the standard sense, but by this slightly more complicated Darwinian mechanism, which I said it inverts only one aspect. It inverts the constraint of resources. When there's excess, now this is possible. I'll just end here by saying a few things about multicellularity. I won't even get to stuff like brains that I'm interested in, but just to sort of give you an idea of how this works and why it's relevant to multicellularity. In multicellularity in our bodies, for example, we start with a single cell or fertilized egg. It divides, makes multiple copies. They carry all the genome with it, all the genes there, but they now begin to communicate to each other, turn each other's genes on and off. In this process, we now have redundancy. Now everything doesn't have to do everything. You don't have to have all the same genes working. And so now what's happened to make multicellularity possible, communication between cells turns genes on and off. As our bodies develop, genes in different cell lines, whether it's going to become heart or brain or skin, some genes are turned on, some genes are turned off, typically by virtue of who are your neighbors, who's far away, because they're sending little signals to each other. But the question is, why is it that multicellularity, you and I are colonies of trillions of cells that all cooperate. Most of them are just going to die out. A few of them, very, very few, are going to reproduce in the next generation, if we're lucky. It turns out that multicellularity developed many, many times. We think now somewhere over 20 times. But there's only 3 groups that are stuck as multicellular organisms. Fungi, like mushrooms, plants, like flowering plants, and animals like us. The key question is, why don't they sort of alternate between being single cell and multicell? The answer is that in each of those groups, the single cell phase, the single cell part of the life cycle, has become parasitic on the multicell part of the life cycle. That is, like the parasites, they haven't lost genes, but what's happened is they've lost autonomy. They allow this process to develop, so they can develop multicellularity from a single cell. An elaborate amount of genes being turned on or off now are accomplishing the same thing to produce an individual of a much larger sort. Let me just end with just a couple of interesting examples here. Lichen, everybody's familiar with lichen, all over the place, very, very successful species, group of species. It turns out that lichens are also a combination. They're all eukaryotic cells with mitochondria in them. But it turns out they're a combination of fungi, bacteria, and various forms of plants. Some of them also have even viruses in them. They're also a combination, but now a combination of a different sort. It's a more social combination. The point I want to make here is that the development of complexity in evolution is the result of this interesting process of overproduction, duplication, in good environments where there's not competition, where you can begin to sample alternatives in which new synergies can be discovered, and then natural selection can stabilize them and make them consistent throughout the lifetime from generation to generation. But in effect, you couldn't have explained this process of complexification just with the Darwinian story. Let me now end by just saying this, that the Darwinian story, natural selection, is about elimination of something. But if you're going to find the right fit between things, you can't just use elimination. You've got to have something that gives you excess that you can sample from. This is the other half of the Darwinian story. The half that Darwin couldn't have thought about because it wasn't possible to even think those terms in his times. But now it is. And now I think what we're going to see as we begin to penetrate into cells and into molecules and into the depth of the complexity of this process, we'll realize that it's a much more complicated thing than Darwin or even our recent philosophers and scientists thinking about Darwinism and thought. As we've discovered how cells work, we've discovered that evolution and development are both much, much more complicated than we thought. So we'll stop at that point. Please.
I just wanted to ask a question about gene degrading and also elimination of something. So what we're seeing is we're seeing our baby fingers getting shorter, genetically as generations go on. And we're looking at also not being born with wisdom teeth. I actually know 2 generations of father-daughter never born with wisdom teeth. Is that an example of gene degrading?
In that case, there is. But it turns out that wisdom teeth, the problem that I haven't gone into, I just hinted at, is that development is a second variant of this whole process. Development has lots of evolution-like processes going on in it. And one of those has to do with assessing your environment. Cells have to not only carry genes that get turned on or off, what genes get turned on or off have to be the result of environmental conditions. And it turns out that teeth are a case in point. Teeth are really sensitive to, first of all, the amount of pressure on them, chewing and so on. The size of the jaw matters a lot. One of the things that's happened in our ancestry is our jaws have become smaller as our faces have become smaller.
Which is why the wisdom teeth are a problem.
That's right. So wisdom teeth
weren't a problem.
They wouldn't have been a problem for our ancestors, not even for the Neanderthals, it turns out.
Thank you.
But certainly for us, it is.
Yes.
But it's not just that the jaws are getting smaller. It turns out that as we're eating softer foods, it turns out there's not enough feedback to get things growing. And so oftentimes those teeth don't grow to their full extent. They stay immature in some respects. Some of them never erupt. So it's a combination. This is why what we call epigenetics, there's all of that communication that's going on between cells and turning on or off genes, is sensitive to the immediate environment. And so in that respect, we have to think about that also as a kind of microevolutionary process going on in our development.
Thank you.
These processes are involved. Way in the back.
Wasn't there a question about epigenetics being transmitted from generation to generation? Is that
Epigenetic transmission.
yeah.
It's become a hot topic, and I think it's a misnomer in part. And I want to clear some of it up. First of all, what we know, one of the first cases that was shown, what was called the Dutch hunger winter. And this was right after the Second World War. And after the Second World War, there was a huge famine in Holland. And women in particular were heavily impacted, particularly pregnant women. And they gave birth to children that didn't do so well, but in fact had a tendency to put on fat. So it was thought that maybe they inherited something like that, an epigenetic inheritance. And then when they began to analyze those children's daughters, the same thing is true. They put on more fat than usual. They had these genes sort of hyperactive. But here's the problem. It looks as though it was transmitted as an acquired character. Acquired because mom was starved. But it turns out that the daughter, who was still gestating, was also being starved. The fetus was being starved. Now, there's a curious thing about the generation of ova in mammals. They're mostly generated before birth. All the ova that will ever be fertilized in any of you women were generated before you were born. And they began to degrade and get lost right at that point. In fact, declines way down well before you even reach puberty. But it turns out that the fetus's ova, the eggs, were also being affected by the hunger. And so what looked like transmission of an acquired character was actually a genetic transmission. It was actually picked up by the developing ova so that the great-granddaughters did not have that effect. Now, there are some other effects that have a stronger transmission, but we think it's basically working that way. It's not that the epigenetic information itself is directly passed on. It's actually marking cells at a very early stage. So, in front of him, you had a question? No?
Did I understand you earlier to say that the two X chromosomes are identical?
No.
Okay.
No. You've got one X chromosome from dad, one X chromosome from mom. However, each of your cells, one of them is shut off. It's what we call a bar body. A complicated way that it's shut off. It turns out a large RNA molecule wraps around it, does funny things to it. So none of those genes get expressed.
And the same one in every cell?
No.
Oh, okay.
It's somewhat random, and it turns out that they get shut off at different stages of development. The most famous example of this is calico cats. Only female calico cats are calico. And the reason for that is that the skin cells, an original progenitor skin cell, has one of them shut off, and one over here doesn't have that same one shut off, has the other one shut off. But now they generate a bunch of other cells, and now you've got a little patch in which one X chromosome is on, and another patch in which a different X chromosome is on. So now you've got this mosaic of X chromosome activity. The reason that women don't have the same problem with color blindness that men do, only have one X chromosome. Yes, only one X chromosome is on in females, but in the retina, the cones are sort of randomly which X chromosome is turned on. And so you get both of those color receptors, which are on the X chromosome, activated. So what's interesting is that using this way to do sex requires that you shut off a whole chromosome. It's another example of the kind of wastefulness. If you produce a lot, you got a redundant copy. Now, there are other ways to produce sexuality, and lots of other species do it in other ways. But this is the only way it would work in us. this is why triple X females, that's one of the possibilities, is to have 3 X chromosomes, do have abnormalities. And if you don't have an X chromosome, you're done. You can't even get started. You need, because it's the most gene-rich chromosome we have. There's more coding genes on the X chromosome than any of the others. So it's a really critical chromosome. So it's a curious case. Yeah.
Hi, I have 2 questions. So following this thread of logic, does that mean women are more sensitive to, for example, diseases that travel on the X chromosome because now they have 2 genetic lines that they might be predisposed to?
Or they're less susceptible because if one of them is not working, the other one's carried something that's useful.
Okay.
And that's why women are less susceptible to color blindness. But clearly, if there is a disease that's carried on the X chromosome, if you're a male, you got it.
Yeah. Okay, good to know. My second question, so taking this paradigm and zooming out from the biological perspective to the kind of socio-ecological.
I was hoping to go there. Good.
So this theory that once you start having a symbiotic relationship with something that essentially takes the burden off of you as an individual being or as a biological being to make vitamin C, et cetera, et cetera. So given how much technology is playing into our lives, you showed the slide with the phone and the
Yeah, I started there, didn't I? Yeah.
It seems like obviously it's going to take a long time to actually develop in this direction, but it seems like what we're doing is we're kind of intentionally building ourselves into a future where we're not only reliant on technology, but we've kind of programmed ourselves so far into technology taking over basic functions that we need to survive, that there's no way out of having technology completely intertwined in our lives.
None of us in this room would work outside of this context significantly if it was degraded to the point where we had to find our own food in the surrounds.
So we're already there.
We're done. But this goes back much further than that. It goes back to the level of the origin, and this is some of my earlier work on the evolution of language itself. Language is something that no individual develops. Language is something that requires that we're part of a social group that share a particular interpretive communicative habit. We have to share it in common or it wouldn't work. It's like vitamin C in that respect. We are now, our whole capacity to think is dependent upon being part of a social group. That means there's also going to be selection maintaining our social stability. Various ways to cooperate are going to be necessary because we can't make it now that we've developed this sensitivity to communicating and thinking this way. It's an incredible tool. The important point of the story that I told from Plato at the very beginning, yes, we've lost our capacity to do lots of memory-like things. But writing has made it possible for me to know what Plato was thinking and possibly what he thought that Socrates was thinking. Writing has done something else. Beyond just spoken language, it's now allowed this knowledge to extend over thousands of years. A lot of my work is dependent, like any scientist, dependent upon knowing the thinking that people were doing thousands of years ago. I'm addicted to that in a much more profound way. It's not a biological addiction in the same sense. And yet, part of my research on studying how brains have evolved in humans is that if you're born into a context in which you don't experience language, in which you're not in a social group, you will become severely disadvantaged. Not just because you can't communicate, but because much of the way your brain works is no longer appropriate. The reason for that is that we come into the world expecting language, expecting being part of a social group, in the same way that we come into the world expecting that there will be vitamin C available to us. We have, in a sense, neurologically de-differentiated to some extent. We're less autonomous than, say, a chimpanzee for that reason. But like the Plato story, we're capable of so much more as a result. It's what we've given up, and it's a spontaneous process. It's like, if I don't have to do it, I'll degrade it. Life is lazy in that respect, just as we are. It's just a natural part of the process.
You started by telling us that we would be studying the evolution of life through natural selection, and not the creation. But do you think, are we getting anywhere near an understanding of the origin of life?
I think so. And now, that would take me a whole other lecture, so I'm not going to do that. But the question is, one of the real challenges and the real reason that Darwin couldn't deal with it, of course, is that the natural selection story assumes life. It assumes reproduction. It assumes duplication of things. It assumes adaptation. And then it just says, if I've got it, now I can get it better. I can readapt, I can be plastic in certain ways. But it doesn't explain how it starts. This is, because it's a side of the Darwinian story that's about what you might call the generative side of the story. How novelty gets generated, how new combinations get generated. It gives us different clues to that question. And so I think indeed there are ways to get at this. But it's not going to be how we thought it was. It's going to be a different story.
Is the story so complex that it might not come for another century, do you think?
No, I don't think so. I think it will come within the next couple of decades. But again, only if we shift off of the old story a little bit and begin to understand the generative side of the problem. Because life begins as a synergy. Parts of every organism depend upon the other parts. They all depend on each other. This is a question about how new synergies arise and how they arise spontaneously. That's the problem of the origins of life. A particular kind of molecular synergy develops. I happen to think that it has nothing to do with RNA or DNA. I think that that's a red herring because we think that since all life that we know of uses that stuff, it had to start that had to be the beginning. But notice that those are just molecules. Copies of molecules are copies of molecules. They're not keeping themselves alive. They're not supporting themselves. They're not adapting to the world. They're just being copied. That's not enough. What's got to start the process has to be a kind of synergy.
When I was thinking about the futurist thought, it's like the cells came from molecules cooperating essentially. And then some of those cells like stuck together and they started depending on each other and they became multicellular organisms. As we organisms start relying on each other, we're it's kind of the same process at another level, and we are part of this superorganism in a way, and it becoming more and more, dependent.
You're right, and of course becoming a superorganism means that the parts lose their autonomy a little bit in order to work better together. We don't like to lose autonomy, and yet we do like to have help. We do like to rely on something that's out there. So whether we like it or not, we tend to give up autonomy easily. Now, we have lots of examples in the world of this sort of thing. If I was to go on for another hour, we'd talk about these examples, like ants and bees, which most of the individuals can't reproduce themselves. And in fact, they work together almost like an organism. We sometimes call them superorganisms. They've given up a lot of autonomy, reproductive autonomy in particular. But in the case of termites, for example, there's the queen, this massive thing that's got a massively enlarged abdomen with lots and lots of eggs being produced. She can't move herself around, so she's got a bunch of nest carriers that move her around and carry her and pull the eggs out when they're ready to come out, and so on. Then there's a bunch of foragers. There's a bunch of individuals that sit at the entrance with giant jaws, these basically soldiers, that they're kind of like different organs. Not quite, they're individuals. They have their own genomes and so on. But like in a multi-cell body, communicative messages have been passed along during development, mostly pheromonal messages that activate, sort of like hormones, turn things on and off in whole organisms, just like hormones turn things on and off in us to change our sexual characteristics. In this case, what this tells us is that sexuality at the level of termites or ants and bees, for example, is more than just two sexes. There's multiple ways that the hormones can change your body for certain functions of the whole, in this case, the whole colony. We're not that way, but we're going in a way similar to that. I'm part of a group that's called Human Energy. If you're interested, look it up on the web. It's a group that's studying what a man named Teilhard de Chardin, he was a Jesuit priest who actually did some evolutionary work. He studied human evolution in Peking and outside of Peking in a cave called Chukutien, where he found was one of the discoverers of what was then called Peking man. But in any case, he felt that the human race was evolving towards what he called a noosphere. Noos is the Greek word for mind. That is analogous to the biosphere, which encircles the earth. There is now, he said, a noosphere, a sphere of ideas and thoughts that encircles the earth. And like a developing organism or the evolving ecosystem, it will become more and more integrated. So he had a view that very much like taking your idea to the next step, that we would become, you might say, a different kind of superorganism. Yeah.
Just a quick observation and a question. The observation is that Darwin, when he said that, all of life has common ancestry, has huge implications. It means that all the bacteria, the insects, the redwood trees, these are our cousins.
Exactly.
We're all related. I mean, and to wrap your mind around that, I think a lot of people, including myself, have a hard time doing that day to day. Anyway, but here's my question, because I really appreciate you incorporating this whole idea about synergy and symbiosis, and just, as an adjunct to the question about technology, we have become separated then from fundamental support systems, our air, our water. I mean, the idea that we have common air, that our water has to serve everybody, the soil and its health is essential for the survival of our species. And yet, when I look around this room, there's not a single plant in here. There's nothing. I mean, our ancestors just a few hundred years ago were living in forests, right, or deserts or wherever. But we've become so separated from that I think, I'm interested in your view on this, we've lost contact with the essential importance of feeling connected to the earth, not just to each other, but the earth itself.
I agree entirely. And I think the real challenge, of course, is that precisely that because we can take advantage of fossil fuels, metals, all the things that we have now available to us, we've become addicted to them. But they have also separated us. At each of those steps, it separates you a little bit farther. Now, I've talked about it in biology as losing information. The genomes get simpler. Why? Because once you're reliant on something else that you don't need to do yourself, then you give up. And as that progresses over time, in this case we're talking about historical time, the human race as a whole has become, as you say, more and more displaced from the earth. Now, part of it has to do with our way of communicating. Our way of communicating, of course, is also abstracted from the world. Language has allowed us to create these ideologies, these illusions, these stories about where we came from or where we didn't come from. I've just told a story here, but it's a story that's, I hope, helping us figure out how to reach back to that piece of the story. But also to understand how we got here as well. We got here by a natural tendency. This is a tendency of all life. To produce as much, to find something that will support you that you don't have to work on. And then give it up and to move on. Of course, we become lazy. We become dependent. And we really feel it when things start to break down. And sadly enough, not realizing by being so displaced from the world, all the poop that we're putting out there in terms of whether it's carbon dioxide or plastic, eventually it's our nest. And you gotta, we used to put it in somebody else's nest, but now we can't.
Right.
Now it's in our nest.
Thank you.
So I was thinking that over 97% of all of the human genome is non-protein coding.
Yeah.
For many, many years, people were calling that like junk DNA, right? And in the last 20 years, we have shown that actually in those non-protein coding DNA, through genome-wide association studies, that's where actually most of the phenotypic variation is explained through genetics. So from that is telling me that those regions that we used to think that were like junk is actually the ones that are regulating when those genes are expressed and in which situations are expressed or not. And it has probably also evolved in the way that you say that multiple duplication events, and now that's why it represents that the majority of the genome instead of just that tiny 3% that is protein coding. Just wonder if you had any.
Oh, there's a lot to be said about that. So first of all, one of the things that we're discovering now is that RNA, the secondary product from DNA, has all kinds of regulative functions all over the body and all over the cell. And in many respects, it's one of the reasons why people have become so excited by what they call the RNA world origin of life story, because it has all of these functions that it can do. As I mentioned, the bar body, the X chromosome that's inactivated, is in part inactivated by a long RNA molecule that wraps around the whole thing. It doesn't produce a protein. We used to think that genes, their only function was to produce proteins. Now we know that probably the main function is to produce RNA. And that RNA can play interfering roles, it can play regulatory roles, it can play editing roles. RNA plays a very significant editing role in what we call spliceosomes, these chunks of our cells that actually take the RNA information and edit it. Our genes they spit out stuff full of noise that gets edited. And it takes a lot of RNA, it takes 5 very distinctive RNA molecules to do the editing, along with actually hundreds of proteins to do it. What we're learning is that the genome is so diverse. But everything I was saying about the genes that I talked about, which are well understood genes, also is relevant to all of that. Now, it doesn't mean that every bit of DNA is playing a regulative role of some kind. There are pseudogenes. There are many pseudogenes. And there are probably many pseudo-regulatory RNA genes. The same process is going to take place throughout. We just don't know how much of it is junk, how much of it is partially useful, how much of it is producing proteins, how much of it is regulating proteins, and there's even some troubling recursive effects that in which proteins regulate genes, which regulate RNA, which regulate proteins in very complicated ways. But this basic principle should be relevant to all of those aspects.
My question is going back to the consideration of what Socrates said about the big change that having an alphabet and being able to write, and instead of memorizing Homer, you could just read it out. And it seems from some of the authors I'm reading that they think that this is what's happening. We're going through a paradigm shift of that sort now with technology and the access to information that we have. Has anybody been studying? I know that some authors are looking, or some scientists are looking at what's going on in the brain and how our brains change in relationship to the languages that we use, and what aspects of languages, which is rather fascinating. Has anyone been looking at that in terms of now, like young people and what they're doing with technology and how their brains might be changing?
There are people looking at that. I don't know much of that research, so I can't really recount it to you. What I do know is that one of the most long-studied features in that respect has to do with what's sometimes called the Flynn effect, which has to do with the fact that IQ scores in industrial societies particularly continually increase. I can't remember how many points per decade. I think it's like, what, 2 or 3 points a decade?
3 points, I think.
And what that means is that IQ tests have to be renormalized. You have to set 100, which is the mean, right? You have to set it a little higher in each generation. Well, it turns out in different parts of the world, that's changing. It's increasing all over the world, but at different rates. It's beginning to level out, I think, in Asian populations now and in some European populations. And other populations are still in the up ramp. Most people now think that that's the result of exposure to media. And that media is actually playing a role in how quickly we consolidate information, how easily we consolidate it, and how quickly we can sort of put it into what we call working memory. This is a process that is probably very generic for brains. IQ is sort of, I think it's spurious in lots of ways. But one of the things that we can say is that if there are some generic functional features that have to do with the way that input gets put into working memory and you can work with multiple things at once, that's very important for almost all those tests. And that looks like one of the things that is affected by exposure to media. Now, how will the new kind of media change this? I don't know.
I noticed that in every human culture in the world that they develop, and women have the choice to reproduce or not, they do reproduce way below the replacement rate. Does this mean that a conscious, viviparous, intelligent mammal is an evolutionary dead end? Or we could say it by growing in the tank, obviously. Or is it just an effect of density in every population density?
So that last part is very important. It turns out that what we call the demographic trend, the demographic transition, has to do with people moving to cities. As people move into cities, first birth is usually later, and birth spacing is usually longer. And that means that in total, there's a slower reproduction rate in general. It doesn't change many other physiological features, but it has to do with choice in various ways. And usually, of course, that requires a choice of two kinds. One way to control sexual access, and another way to control insemination and eventually fertilization. So obviously birth controls of various forms play a role in this. Outside of the cities, reproduction rate is typically considerably higher than replacement rate. But that's because infant mortality is very high. And so we don't know quite why this is happening, why it is that as people move into cities with higher density, but also with more support, that they slow their reproduction. And there's a whole bunch of factors. Education is a factor as well, it turns out. But this is worldwide. This is not just in the West. So one of the reasons that we can predict that the Earth's population, if we let everything continue as it is, will probably plateau somewhere around 2050. Because there's more and more movement to these high density areas. And we're seeing the reproductive rate drop significantly as this happens. What we have not seen, the reason it will continue to rise, is because longevity has been increased. And so that will also plateau at some point, we think probably around 2050 as well. And then we'll begin to see a slow decline in the Earth's population, assuming a whole lot of things. But this is just basically a demographic trend that's pretty clear. Why it's happening, we're not sure, but we know what the correlates are.
Terry, you mentioned a leveling off, a plateauing of intelligence measurements in Europe, countries of Europe and in Asia. Tell me, the United States hasn't plateaued. Can we do better?
So what I can tell you is that different populations in the United States are at different places in their rise. So if you think about it, it's a rise sort of like this, and then we reach a peak. I think that there's a level at which you can't speed it up anymore. And what we find is that different subpopulations are in different places along that curve. The Asian American population is already much further along than the Caucasian population in that respect. there is a look at different populations, the Hispanic population, African American population, and so on. There are different stages in that, but also different countries as a whole are in different places along that line. So it's, I think what it will show us is that there is a place, a maximum we can go in terms of IQ testing. Whatever IQ is actually finding out. I think it's actually finding out some fairly trivial general things about how the brain works. But nevertheless, I think there's a plateau to be found. We're not there yet, but we're darn close.
Thank you. Before we give Dr. Deacon a final round of applause, I ask you to first remember what happens in 2 weeks. Right here, it's Hacking Gerrymandering with Dr. Ellen Bayomet. And as you give this final round of applause, remember to thank yourselves for being here and asking such great questions. Thank you for being a great audience. Remember to thank a little bit of your applauses for Hot Monk Tavern for hosting us here. And now maybe, how about a final round of applause? Dr. Terry Deacon.